Hello everybody, my name is Daniele Raimondi and I am a postdoctoral researcher at KU Leuven. Today I will talk about a paper we recently published about the end-to-end multitask prediction of quantitative traits in Arabidopsis thaliana. Genome interpretation is the umbrella term describing the scientific endeavor towards understanding the genotype to phenotype relationship. Being able to precisely model how the information encoded in our genome leads to the observed phenotype would indeed constitute a crucial advancement for genetics and molecular biology, and could open a new era for precision medicine and agricultural technology. Early efforts towards genome interpretation included statistical models for phenotype prediction, such as polygenic rescores in humans and the Bloop family of methods for crop and cattle breeding. Nevertheless, those approaches were developed before the next generation sequencing explosion, and they all relied on SNP array data that are normally used for genome-wide association studies. SNP array data do not sample every position in our genome or exome, but they just take a sparse snapshot, something like this. To be more accurate, it would actually look something like this. As you can see, it is quite difficult to get a real idea of what's going on from such a sparse picture, and indeed, SNP array data is unlikely to contain the truly causative variants. The hope is generally that some of the observed SNPs are in linkage disequilibrium with the truly causative ones. Whole genome and whole exome sequencing, on the other hand, can theoretically identify every form of genetic variance with respect to a reference genome, and this data is indeed much more likely to contain causative variants for the phenotypes under study. Now that we have all this genomic data at hand, we unfortunately discovered that the bottleneck just shifted from a data availability issue to a data interpretation issue, since causative variants are still hidden in the plethora of neutral variants regulating normal phenotypic expression. And this is leading towards the proverbial needle into the haystack problem. Moreover, genomes are large and partially structured data that is not easy to feed into conventional machine learning methods and pipelines. Our answer to those challenges is based on three tenets. First, the big data size and the peculiarities of genomic data means that they require the development of ad hoc neural network architectures able to accommodate them. Second, those models should follow a data-driven approach in which inheritance patterns emerge from the data. And third, we assume that the genotype-phenotype relationship is highly nonlinear for most of the non-trivial traits. In this study, we focused on Arabidopsis thaliana, which is a well-studied small flowering plant that belongs to the Brassicaceae family. It has a small genome, around 125 megabases, which is divided into 5 chromosomes, but it does a number of genes that is comparable with humans, around 27,000. We retrieve 1,000 publicly available genomes of Arabidopsis thaliana from 46 countries, and we associated them with 288 real-valued phenotypes from the Arapheno database. We then developed a neural network model that we called thaliana, which takes as input a compressed description of the gene mutational load carried by each individual's gene. This gene-centric sample description is parsed by a shared model, G, in order to minimize the overall number of parameters in our model. And this module G produces a minimal latent representation for each sample. This representation is used by the final multitask layer P to concurrently predict all the 288 phenotypes. To give an idea, those are the scatter plots of four predicted phenotypes obtained in cross-validation. In clockwise order, we have leaf number, root length, fruit number, and the days to flowering time. Overall, thaliana predicts 75 of the 288 phenotypes with a Pearson correlation greater than 0.4. To show that our genome-wide multitask approach is beneficial with respect to conventional approaches, here we compared thaliana results with phenotype-specific models that used only the known associated genes to predict the 35 phenotypes for which this kind of data was available. In 77% of the cases, thaliana obtained better performances with respect to those phenotype-specific models, indicating that our multitask end-to-end genomes in, phenotype out approach can improve over conventional methods. This work has been developed in collaboration with my colleagues at KU Leuven, at the University of Torino, and in France. This presentation provides a very brief summary of our work, and if you're interested, I invite you to find the full paper at this QR code. Thanks a lot for your attention, and I wish you a very nice day.